biodiversity of the living world is affected by the interaction between ecological and evolutionary processes. Ecological adaptation by natural selection can play an important role in the origin of biodiversity. It's this adaptive divergence to different environmental conditions that can transform the variation we see within populations to the differences we observe between taxa. So in addition, coevolutionary interactions can enhance the effects of natural selection to create biodiversity. And specifically, the effects of adaptive radiation can ripple throughout a community, and it can provide the template necessary for population divergence of associated organisms. And so it's this idea that biodiversity in certain situations can beget further biodiversity, and organisms can sequentially radiate in concert. So competition is an ecological process that can promote speciation. And intraspecific competition is an incredibly important component of many models of speciation, particularly sympatric models. One of the first models was developed by Rosenzweig in 1978. In his model, the frequency-dependent effects of intraspecific competition create a fitness landscape where phenotypes that use the extreme portions of the resource distribution can experience increased fitness over those intermediate phenotypes. So really what this does is it creates a bimodal distribution of phenotypes that can evolve into subpopulations, and if these subpopulations experience some form of reproductive isolation, this can lead to ecological sympatric speciation. So this initial model was kind of the impetus for an explosion of intraspecific competition models in the literature. So while it's less well studied, interspecific competition can promote speciation as well. So similar to intraspecific competition, if different species share a trait or a phenotype that will allow them to overlap in resource acquisition, a limited and shared resource, competition will occur, and this can lead to the evolution of ecological character displacement, where species will use different portions of the resource distribution in areas where those species overlap, but in areas where those species do not overlap, the displacement will not occur. And so in this study, we wanted to test for the role of intraspecific competition and ecological character displacement and this sequential divergence idea using the apple magotfly Rhagoletis pomonella and a community of endoparasitic specialist wasps that attacked this fly. I'll refer to these parasitoids by their respective genera names, Eutetes, Diachasma, and Diacadmymorpha for the rest of this talk. So the apple magotfly is this model system for ecological sympatric speciation. And there's a species complex of this fly that's composed of taxa at varying stages of genetic divergence along the continuum. This includes the textbook example of sympatric speciation, the apple and hawthorn host races of the fly. And so these flies lay their eggs into fruits of different host plants. And more or less, these flies are exchanging genes at a reduced but detectable level. Another important component here is that all of these plants geographically overlap, and they all fruit at different times. And so an important component that reduces gene flow between these diverging populations is that these flies emerge after an overwintered diapause to coincide with the fruiting phenology of the respective host plants, and all of these host plants fruit at different times. And given that the fly doesn't live very long, about 30 days in nature, this can actually create rather strong prezygotic reproductive isolation in this system. So we previously showed that as the flies diverge and switch to new host plant habitats, this creates opportunities for this entire community of parasitic wasps that are attacking the flies to diverge in parallel. And so it's this idea of multiplicative sequential divergence. As one fly shifts to a new host plant, the entire community diverges in parallel. And so we found that the same host-related ecological adaptations that reduce gene flow between these fly populations, that is, differences in the time when they eclose in nature, are the same ecological adaptations that drive the reduced gene flow between these wasp species as well. And so the impetus from this current study came from two observations. The first observation is that if we look within a host plant, it appears that different species of wasps are emerging at different times, suggesting that they could be temporally subdividing the host fly resource. The second comes from a natural history observation, a rather old one, and this suggests that multiple species of parasitic wasps can begin development in the same fly, and if this resource is limited, this could suggest that they might be under fairly intense interspecific competition, larval parasitoids at this level. So that's what that would look like. So this is so we wanted to test a temporal variant of the ecological character displacement idea, and we wanted to test a multiple habitat sympatric model with gene flow and test for the role of interspecific competition in resource subdivision and sympatric divergence in this WASP community. So here's the model we want to test. These large smooth distributions represent the fly niches. You can think of this as apple and this is hall. These dashed distributions, the smaller ones, represent the wasps using this distribution. And so in the absence of interspecific competition, we can see that there's actually a measure of reproductive isolation between wasps using different fly niches, just based on the timing of when these wasps can attack these different flies in nature. But if interspecific competition is occurring, the wasps are subdividing this resource and it's allowing them to coexist, then wasps on different hosts may experience increased allochronic isolation in the presence of interspecific competition compared to the absence, that's this overlap, versus this overlap here. So it could be the interspecific competition, or more specifically, the alleviation of competition that allows these wasps to temporally subdivide this resource, and that increases reproductive isolation among wasps using these different fly niches. So we took a five-pronged approach to address this question. I'm going to go through each one of these in turn. So first, we went out in nature and we sampled parasitized flies for 10 years across 5 different sites for apple, hawthorn, and blueberry fly hosts. We found 2 really rather interesting patterns. The first is that all 3 species exist on all the hosts in nature, with the exception of Eutetes, which is not present on blueberry. This implies that these populations populations of these wasps stably coexist across space and time. And then we see another interesting pattern where Diachasma across all sites and all years is the most common parasitoid, suggesting that if they're interspecifically competing, it's a superior competitor. Next, we reared about 2,000 pupae individually, and we wanted to see what came out of them. And so there's a couple results that came out of here. The first is that no more than 1 wasp will come out of a single fly pupal case, suggesting that if they're interspecifically competing on that level, multiple species may go in, but only 1 will come out. Second thing is that for pupal cases where wasps came out of, we simply looked inside of them and asked the question, are there any resources left over? Limiting resources is an important component of interspecific competition, and we didn't find the presence of any resources left over, so this suggests that this resource is limiting. So next, what we did is we preserved flies from various stages, beginning at the larval stage all the way to 20 days post-puparium formation, and then we developed genetic DNA diagnostic markers that would amplify PCR products specific to each one of the 3 parasitoids that we're working with. And so a couple of interesting results came out of here. First, we found that multiparasitism is common on the individual fly level. It varies between hosts, ranging from about 28% in blueberry all the way up to 50% or 60% in apples. So again, this means that multiple species, larval parasitoids, can begin development in the same fly. And so here's what we did next. I'll walk you through this graph. This y-axis here is percent parasitism. X-axis here are these fly life stages. And we see an interesting pattern. For Diachasma, we see that parasitism stays relatively constant as flies get older, but for Diachasmomorpha and Eutetes, their parasitism rates decrease as flies get older. So what this says is at the beginning, multiple species are beginning development inside a single fly pupa, but as time increases, Diachasma develops and starts consuming these interspecific competitors, and actually it wins out. And we see this pattern across all 3 hosts, apple, hawthorn, and blueberry. So next we actually wanted to ask the question, well, what happens when we remove the replicates where Diachasma is present, and just those replicates where Eutetes and Diachasmomorpha are present? Is one of those a superior competitor? And we actually see a similar pattern where Eutetes is out-competing Diachasmomorpha. And so what this suggests is that there's this hierarchy of competitive dominance, where Diachasma is the most competitively superior, followed by Eutetes and Diachasmomorpha. So next we wanted to actually look in nature for the temporal resource partitioning of these wasps using these different fly hosts. So I'll remind you, this is the fruiting phenology of the respective host plants. Blueberry fruits earlier than apple, which fruits earlier than hawthorn. And then when we look at the eclosion times, standard eclosion times in the lab, we see a similar thing where blueberry flies emerge earlier than apple flies, hawthorn flies. And then again, we see this temporal resource subdivision of these wasps on these respective host plants With here, Eutetes emerging on apple emerging earlier than Diachasmomorpha emerging earlier than Diachasma, again suggesting that they're temporally subdividing this resource. So this is one component of the life cycle, but we wanted to look at two other components. So we went out and just performed sweep nets at various times of the year to see when these parasitoids were active. And we also collected fruits at different times of the year to see if they were parasitizing flies inside of fruits at different times of the year. And we see a very similar pattern at these two other components of their life cycle. Here, timing of parasitism and timing of adult activity, where there seems to be temporal resource partitioning of species of wasps within a host. Here's another example where timing of parasitism, Eutetes is the earliest, followed by Diachasmomorpha, followed by Diachasma. So what we can do is we can take any pairwise comparison of these wasps attacking different fly hosts, and we can actually calculate reproductive isolation via this temporal subdivision in the presence of interspecific competition and in the absence of interspecific competition, and we can actually test our model. And so I'm going to graphically show you what these results look like for just a single comparison, but you can do it for any one of these actual experiments we performed and any pairwise combination. I'm going to focus on eclosion time for apple and hawthorn wasp communities. So this is what these results look like. Again, these smooth distributions represent the fly niche, and this is actual real data of eclosion time overlap between green apple wasps and red hawthorn wasps. And so their distributions overlap about 82% in nature. So we can see there is a measurable amount of reproductive isolation between the entire wasp community. These curves under here represent the entire wasp community. There is a measure of reproductive isolation just because these wasps are attacking flies at different times in nature. But if we actually look after interspecific competition and resource subdivision, we can see that the overlap is reduced for all three species, from 82% down to 50% for Eutetes, 82 to 62 for Diachasmomorpha, and 82 to 45%. So as they temporally subdivide this resource within a host, it actually increases reproductive isolation of the same species of wasps that are diverging on different fly hosts. And we see this across the board for all the different pairwise comparisons and all the different experiments. In every single case, reproductive isolation in the absence of interspecific competition is much lower than it is in the presence of interspecific competition. So what did we find? We found that species stably coexist across space and time. It appears that their resources are limited. This is a critical component of competition. We now know that there's multi-parasitism. Multiple species of wasps can begin development in the same fly. Therefore, they're competing for this limited resource. And it seems that at multiple stages on their life cycle, they're temporally subdividing this resource. And there's this competitive hierarchy as they're competing, with one species being much more dominant than the other two. And it's this temporal subdivision of these resources that allows for these populations to stably coexist. And it's this stable coexistence that actually provides the template that's necessary for sequential speciation to proceed, but specifically the multiplicative properties of sequential speciation to proceed, because you can't multiplicatively amplify biodiversity across trophic levels from one fly to multiple species of parasitoids unless you can get those parasitoids to coexist. And so, all in all, this temporal subdivision increases reproductive isolation between these diverging populations and again promotes sequential divergence. Number of people helped out on this project, too many people to thank, really. I'm forever in debt to them, and of course funding sources. And am I out of time? All right. Thank you, everybody.